For more than 30 years, Dr. Marcus Peter has immersed himself in cancer research, most recently at the Robert H. Lurie Comprehensive Cancer Center of Northwestern University, pioneering studies in the field of cell death research and developing strategies to selectively kill cancer cells. He is now using the same trailblazing methods he has uncovered in cancer research to lay the foundation for a new avenue of treating Alzheimer's disease and potentially other neurodegenerative diseases. He is the Tom D. Spies Professor of Cancer Metabolism and a professor of medicine in the Division of Hematology and Oncology here at Feinberg, and joins me today to discuss his research. Welcome to the show.
Thanks, Erin, and thanks so much for having me.
So let's start off by talking about cell death research. As I mentioned, this is something you've been doing for about 30 years. Explain this research to me.
So how can we make cells die? Obviously, there's one particular type of cells that we'd like to kill, the cancer cells. So for the last 30 years, basically, it's all been about cancer research and how we can tackle this. How can we overcome resistance to therapy? How can we eliminate cancer cells without affecting normal cells? And it started out with one particular form of cell death that's called apoptosis. It's one of those regulated forms of cell death. And we studied that and we deciphered how it works. And I learned a lot about make cells die. However, more recently, last 10 years or so, I realized is that as a biologist, if I was Mother Nature, why would I put all my money on one form of cell death only if I wanted to eliminate cancer cells, which is critical for survival, right? And in the early 2000s, pretty much all cancer centers worldwide had one mission, that was to cure all cancers by 2015. That's 9 years ago. So out of that frustration a little bit arose a driving force, the desire to start over and think about what could it be that has made us survive as multicellular organisms the last 2 billion years, basically. And then about 14 years ago, we stumbled over something that allowed us to kill all cancer cells in a way they could never become resistant to this day. And we now know when we do this, when we trigger this mechanism that we're going to talk about today, then we also don't have any measurable effect on normal tissues yet. That's not to say if it ever gets into patients, we won't have side effects, but so far we don't see anything in animals. So that's how it started. So initially it's nothing to do with neurodegenerative diseases such as Alzheimer's disease, but it was this mechanism. And so we started diving into how does this work? It is based on short RNAs.
And you have found that these short RNAs, in particular non-coding RNAs, play an important role in regulating gene expression and could have implications for cellular function, survival, and potential therapeutic strategies. Tell me more about this.
Gladly. So we have a number of large molecules in cells we call macromolecules. And they are these different classes, and pretty much everybody knows about DNA. And then there's protein. Proteins basically is what makes all cells function properly. And in between are certain types of RNA that code for those proteins. So the DNA is converted into these long RNAs. The RNAs give rise to the proteins that makes the cells function and survive. It's important in this context. But then there are short RNAs, and they don't code for proteins. So we call them non-coding RNAs, and they have multiple functions. And a class of those short RNAs, they're only 20 nucleotides long. These are the building blocks of DNA and RNA. These short ones, they actually negatively regulate gene expression. So basically, they target the long RNAs. The moment the short RNA, which is sometimes a piece of the long RNA, targets the long RNA gets eliminated, the protein can no longer be made, and that will have effects on cells. We can induce this process. It's called RNA interference, or RNAi. But nature, of course, invented it and does it much better. And there are professional short RNAs who do this, and they're called microRNAs. And microRNAs do target dozens, sometimes hundreds of these genes in networks, and they do maintain differentiation. They do a lot of different functions.
Thank you for that explanation. Can you tell me more about what makes some of these short RNAs toxic?
To understand why certain short RNAs can be toxic, we need to understand that we have about 20,000 genes in our genome, roughly. And about, again, roughly 10% of those genes are required for survival of all cells. Of those 20,000 genes, many of them can be eliminated. You may have some effects, some response by the cells, but it's not critical. And then there is a subset of about 10%, about 2,000 of those genes, And they're defined by taking one of them out in a cell and every cell would die. And we call those essential survival genes. Now think about it. If there's a long RNA that codes for one of those critical survival genes, what would happen if we eliminated or target that long RNA? The cells would die. So that means that over the last 800 million years, at least, that's how old microRNAs are, all the way from little worms to us, that there must have been an evolution to prevent microRNAs from ever targeting, attacking the mRNAs that code for those critical survival genes. And that is exactly what we found. And since the way these short RNA target the RNAs is only with a minimum of 6 nucleotides, it's a very short region that we call the seed sequence. So if that seed sequence has a certain nucleotide composition, we have 4 nucleotides in all of nature, and if it has a certain G richness, as we say, but only in this short stretch, what we call the seed, and we put that in any short RNA, and it does its thing and targeting the long RNAs, every cell would die. So that's why not a single highly expressed microRNAs we have in every cell, or almost all cells in our bodies, carries this code we call the kill code, because every cell would die. Now, if you really believe in our hypothesis, if you buy into that it is that old and that could be a mechanism, that would be, of course, a fantastic way for nature not only to protect us, but also kill cells that are unwanted. So in fact, this would be the ideal anti-cancer mechanism if you could control it, because obviously it is very dangerous. It's very powerful and need to be controlled tightly. And coming back to what I said initially, it does activate multiple cell death pathways. We have about 20, 30 of them today, not just 1 or 2 or 3 as a few decades ago. So now you have it. So you can activate all those in parallel. And you can imagine once you're attacking dozens or hundreds of genes and these targeted sites in these genes are even more, how can a cancer cell ever become resistant to that? It would have to mutate basically all these genes, eliminate all these sites. So that is the mechanism that we believe we discovered. We call death induced by survival gene elimination, in short, DICE.
Tell me how you discovered that these toxic RNAs allow cancer cells to die, but not normal cells.
When we realized that we have certain types of short RNAs that are toxic to pretty much all cells and were super toxic in killing cancer cells, we then started delivering those constructs to mice, to rodents in cancer models. And yes, we had all expectations that those mice would drop dead after the injection, but they didn't. They're perfectly fine to this day. So it turns out that, at least from what we can tell, every tissue we looked at, the liver being the most important one in this case, is unaffected, which raises the question, since those survival genes you would think would be expressed in all cells, like all of us have a heart and a liver, why would normal cells be spared? And then it hit us. It was basically the fact that microRNAs are expressed in all cells, and microRNAs are required to maintain differentiation and maintain all of what we are. And when you look into what cancers do, they're the opposite. They are de-differentiated. Cancer, for the most part, is not really killing people. It's metastasis. 90% of our deaths occur through metastasizing cancers. Metastasizing cancer are the opposite of a differentiated tissue. So then, if you believe that microRNAs maintain differentiation, then in order to become a really good, mobile, highly killing cancer cell, you need to get rid of those microRNAs. And that's exactly what we found. It's also been published by others before. There's a global downregulation of all microRNAs in all human cancers, and some of the cancers they studied in rodents as well. And that kind of opens up the machinery in the cell that mediates RNAi in a simplified way. And then the other aspect is that those survival genes, they're not only good for survival of normal cells. Cancer cells are under a lot of stress. In order to resist that, to become more resilient, guess what? They do upregulate all those survival genes that we are targeting with our therapy. So there you have it. You have a complex of proteins that mediates RNAi that is open because the protective microRNA is no longer there. And you have more of the targets that we're targeting. And that allows the cancer cells to die and normal cells not. And that was exactly the question, what would happen if normal cells would lose their protection? Again, known for many years, there is a downregulation of microRNAs in many tissues in mammals, including humans, with age. So there is an aging connection.
So why does this mechanism exist, and why are normal cells protected?
I think we asked ourselves, it's great that we can treat mice and we can have an effect on tumors in these mice with no effect on normal cells. But why? I mean, there's an evolutionary underpinning. There's a rationale why this exists. If this were a mechanism that would be effective in every single cell, hardwired into the cell, it would always be limited to a cell. The selectiveness between a cancer cell and a normal cell would just be at the level of what makes a cancer cell different from a normal cell. It would not explain why a normal cell is resistant to this mechanism. So there had to be something, and I believe we found it. And that is, there are endogenous RNAs in the cells that have this code, and they are needed. And these are long RNAs. And many of the long RNAs, they in fact, the majority of all RNAs in the cell are so-called ribosomal RNAs for the aficionados, or tRNAs. And they are structural RNAs. That means they're not really regulatory, they're structural, and they have this specific nucleotide composition. It's been known for many years that they can be degraded into small RNAs, and since they are so hugely abundant, they can sneak into this RISC complex, and they could potentially kill cells. And that's also known, that they get degraded and enter this complex and mediate RNAi. So we now believe, and that's how nature works, that those microRNAs are not only maintaining differentiation, and they're very highly expressed in all differentiated cells, but they also protect us from any endogenous toxic RNAs. They're not just these microRNAs, they're also the fragments of these hugely abundant RNAs that contain the kill code. So that, I think, is why it evolved this way. And fun fact, that's also known for many years, that these, again, for the aficionados, tRNAs are highly increased in Alzheimer patients and when we age. It's all published. And you put this all together and said, wow, that's what we need to protect her for. And that's why we now come in with exogenous sources, basically coming from the outside with the therapeutics. That's why normal cells are still protected, because they're still busy protecting themselves from themselves, basically.
Are there other folks around the world who are also studying this? This is something that your lab has discovered, and now that you're bringing into other diseases. But tell me about around the world. Have other folks started to look into this type of science for cancer treatment?
To this day, we have published 21 papers on this in multiple diseases in major journals, and no, to this day, not a single group worldwide has picked this up, but one. There's one group in Kentucky that 4 years after original discovery traced our steps and did exactly the same that we found and published it in the context of prostate cancer. To this day, to my knowledge, this is the only paper that ever followed our path. But that's fine, because we just keep developing it.
Well, that leads me to this next question, which is, you're continuing on this journey through cancer research, and then there's this pivot to Alzheimer's research. How does that come about?
It actually was my wife who's in the group, Andrea Merman, who figured, well, if you're right and there's this death-inducing anti-cancer mechanism, shouldn't there be a case where this is overactive, and shouldn't that result in some sort of disease because of loss of tissue, such as neurodegenerative diseases, for instance? And said, hey, that's a good idea. And since this is an anti-cancer mechanism, we figured, well, then we would postulate that these patients may have a neurodegenerative disease, but they may have less cancer, because the anti-cancer mechanism would be active in every cell in the body, right? And so she started searching, and the first thing found was a disease called Huntington's disease. Nice thing about Huntington's disease, in contrast to Alzheimer's disease, has a single cause. There's one gene affected, Huntington, by an amplification of a certain sequence in nucleotides. And there is evidence in the literature that these sequences can act like short RNAs, and they're uploaded into the complex that mediates RNAi and regulate genes just like microRNAs. And when we realized that, Andy ordered short RNAs and tested whether we can kill cancer cells. And boy, could we kill cancer cells. This was 10 to 100 times more potent than the original ones, the dice-inducing ones. And so we looked into Huntington's disease. And indeed, Huntington's disease patients, there are multiple studies, corrected for age and everything, that showed they have 50 to 80% reduced cancers. And that's all cancers. Wow, we thought, maybe we should start looking into that. So that's one of the reagents we're developing as a new form of cancer therapy, these Huntington-derived super toxic RNAs. Well, Huntington patients have decades of symptom-free life, and then there is an onset and it kicks in, and then it goes downhill from there. And that is also true for pretty much all those neurodegenerative diseases, from Parkinson to ALS to Alzheimer's disease. And since here at Northwestern we have these fabulous collaborators, I teamed up with the group Bob Wasser at the Mesolum Center for Alzheimer Research here at Northwestern. And we started looking into this. And then we tested multiple models, mouse models, neuron-derived cell line models treated with the specific toxic proteins that are believed to be the cause of Alzheimer, induced pluripotent stem cells differentiated into neurons, and something we're going to talk about, I guess, also later, the superagers. And when we put all this together, we realized, indeed, these toxic RNAs accumulate in the cells in this complex that mediates RNAi in Alzheimer's patients. In Alzheimer, indeed, late-stage Alzheimer patients get much less cancer. And funny enough, late-stage cancer patients don't get dementia that much. So there is this inverse correlation. And once we did the literature search, we found the same connection to less cancer for Parkinson, for ALS, for all these diseases. That's why we got interested in Alzheimer's disease.
So now we're going to dive into the current study that we're talking about today, recently published in Nature Communications. Tell me about this study, some of the results, and what you were able to publish.
Yeah, this study actually was based on a previous study in the context of ovarian cancer, where we looked at patient material and mouse models, and we realized not only can we kill cancer cells with these toxic RNAs, but when the cells become resistant to chemotherapy, in ovarian cancer, that would be mostly platinum-based chemotherapy, then the ratio of the non-toxic to the toxic short RNAs in this protein complex in the cell that mediates RNAi, it's called the RISC complex, by the way, changes. So in the resistant ones, you have more of the non-toxic ones, providing a better protection against toxic ones. And in 2 previous papers, including this one, we showed that chemotherapy, to some extent, unleashes a wave of toxic short RNAs and in part kills cancer cells through this mechanism. So we kind of knew that the ratio of the non-toxic versus the toxic ones in this RISC complex that may predict treatment outcome, or in other words, may predict whether any tissue would be susceptible to this or not. And then we had to develop this tool that allows us in a very standardized fashion to analyze the content of this protein complex that mediates RNAi and plot it and quantify it. And so once we had that and validated it, we then started establishing mouse models, different types of mouse models that mimic the human disease Alzheimer's. We got cell lines that were treated with the toxic proteins that everybody else studies, which are, of course, on top of this cascade that leads to the neurotoxicity seen in the disease. And we looked in other components, and we got, as I mentioned, cells that derived from stem cells, turned into neurons, and we got those actually from Alzheimer patients and studied them. And in all these cases, we found that in the context of Alzheimer, the amount of the toxic seed-containing RNAs in this complex is higher than the non-toxic, or the ratio shifts, similar to the study on the drug-resistant ovarian cancer patient. It shifted. So they become more susceptible to DICE, this mechanism we discovered. And when we treated the cells acutely with this toxic protein that we know induces Alzheimer's disease in patients, then that resulted in a very clear dip of the non-toxic, the protective microRNAs. That's why we call them protective microRNAs. There's a clear correlation between your ability to deal with the stress and having the protection.
You were able to leverage these findings from your study and apply what you found to the brains of superagers. Now, we have done previous episodes about superagers and the superaging research program here at Northwestern. It recruits and studies adults over the age of 80 who have memory capacity of individuals who are at least 3 decades younger. And after they pass away, superagers donate their brains to the research program to help investigators understand what's going right with aging as opposed to what is going wrong. So tell me what you found when you investigated your theory on the brains of these superagers.
It was a very preliminary study, but we got 3 brains of these superagers and subjected them to the same type of analysis and compared them to regular folks like us, right? The non-superagers. And turns out, indeed, as predicted, the ratio in those brains of the toxic to non-toxic one was towards the non-toxic. They still have more protective ones. So I think we hypothesize at least, that these people, for reasons that are unknown, aging more slowly, which of course gets us to where we need to take this whole thing, right? We need to find a way to reduce the loss, to slow down the loss. We'll probably never be able to reduce it, but to slow down the loss of the protective microRNAs in a number of tissues, selectively, hopefully in the brain. And that should help with basically make us more resilient towards getting any of those neurodegenerative diseases, or may even be helping with aging.
But there's also this idea out here that this study could lead to new treatments for Alzheimer's, which is desperately needed. Can you talk about that aspect a little bit and sort of a pathway forward for potential new treatments for Alzheimer's?
Absolutely. That, of course, is exactly the flip side of our cancer approach. In cancer, of course, we're trying to design the most toxic short RNAs that have the highest selectivity for cancer cells and not affecting normal cells. Now, in Alzheimer, it's exactly the opposite. Here, we want to either prevent the toxic RNAs, if they have a major contribution to the disease, to do their thing, or we would like to increase the amount of the protective ones to make it more resilient and maybe push the onset of Alzheimer from, your early 80s to your early 90s. And then it may not be that much of a problem anymore. And this is, I think, could be feasible, but requires a lot of basic science work, because what we need to study now, and there are some ideas and some data out there, is what are the mechanisms that regulate this? What is the mechanism that regulates the ratio between the toxic and the non-toxic one? What's the specific about the non-toxic ones, and how could you elevate that in cells? And that is a biological mechanism that has not been discovered, but there are some ideas, and we have some ideas of screenings that we could set up, design certain screens to screen for small molecules that could do that. There's actually an antibiotic called enoxacin that has been shown in mice, not with Alzheimer, but with ALS, a model for ALS, to alleviate pretty much all symptoms in 2 different models. And this thing is known to stabilize microRNAs, in our book, protective microRNAs. So there is already a drug out there which was never screened for, was never designed to do that. And I'm sure we can do better once we know what we're looking for, to design specific screens to get small molecules that increase the number of protective ones. I think that would be my favorite approach, because I think it may have the least side effects rather than, playing with the toxic ones. And that's the whole thing. Treatment is one issue. Prevention is another. Obviously, the safety profile for anything that you want to use for prevention is very different, because you want people to take this for decades. So you better have no side effects whatsoever. But if you're in an acute situation, like somebody has advanced cancer or advanced Alzheimer, you can have a beneficial effect with something that has a less favorable safety profile. And so these are 2 different avenues, which hopefully will be explored. And we can't do this alone. We're a small group. We need the help of the community to buy into this, start looking into that, and some people that are way smarter than us coming up with ideas to make this happen.
So if you could wrap this up for me today, what would you like people to know about your lab and what's next?
What's next in the cancer arena is that even though this is by design a pan-cancer mechanism, that's not to say that there may not be toxic RNAs that are particularly active in one or another cancer. So we need to get the full landscape of what are the endogenous short RNAs. As a basic scientist, I would like to continue to make discoveries in this area and figure out how it's regulated. That's the next big step, and how it works to be able to use it in a better way to treat diseases. And of course, again, all depending on funding and opportunities, we have no issue in venturing out into other diseases. We did have a paper last year in Journal of Virology, and the first evidence how viruses, in this case HIV-1, kills infected cells using the DICE mechanism, because if it's 1 billion years old, it has to be everywhere. And so obviously we are a single, very small lab. We cannot study everything, so we have to focus.
I understand you've also started a company to help move this forward into the translational science field.
The name of a company says it all. NewArgo, which is NU for Northwestern University, or NEW if you want. And Argo is, of course, for Chicago, or for the key proteins, they're called the AGO proteins, that are part of this complex that mediates RNA interference. There you have it. NewArgo is not specifically, it's in the name, a cancer-fighting company only. But we'll see what happens. We will be able to sponsor some of the research, at least initially, in my lab here at Northwestern through the company that should start hopefully this year. And then obviously, who knows where this will take us.
And I just have one more question. What advice would you give to younger investigators who are listening to this and they're realizing, wow, you had to forge your own path and take a lot of risks and maybe do things that weren't the popular thing to study or do? What advice do you have for folks who that may sound a little intimidating to them? And what advice would you give to a younger person just starting off on this journey?
All depends on your level of risk tolerance, right? So you have to develop something that is grounded on what others are doing, because you're always, as we say, standing on the shoulders of giants, and you have to take off from their shoulders. Then you really need to make sure that you make a name, you carve out a niche, and you make a career. In order to get a grant, a federal grant, which is the only thing that counts at institutions like ours, don't find what proves your hypothesis. Always try to disprove your own hypothesis. This thing started out by a phenomenon we couldn't explain. And when I realized what it is, I challenged everybody in the lab, disprove me, show me that it is not true. And every time we dove into further, we found another mosaic piece that fit into something that turned into this whole concept. I think that's the advice for young people. Go your own way, but initially have your bread and butter project, play safe, make a name, get the funding, but then don't care about anybody.
Thank you, Dr. Peter, for coming on the show. You have some very exciting things in the pipeline. We'll continue to follow along. So thank you.
Thank you so much for having me. This was fun.
Thanks for listening, and be sure to subscribe to this show on Apple Podcasts or wherever you listen to podcasts, and rate and review us. Also, for medical professionals, this episode of Breakthroughs is available for CME credit. Go to our website, feinberg.northwestern.edu, and search CME.